Good morning, students. So we are done with the body fluid introduction part, and now we'll come back to the very important part of the topic, that is formation of blood cells. So we'll see now how this form element of the bloods are formed. So it is a process by which the form element of the blood develop, and it is called as a hemopoiesis, or it is also called as a hematopoiesis. So now the process by which this form element, which include WBCs, RBC, platelets, which are formed, this process is called hemopoiesis. So what is the site where this takes place? It's a red bone marrow. It is a highly vascularized connective tissue which is located between the trabeculae of the spongy bone tissue. So now we have already seen what are the spongy and compact bone tissue are. So this hemopoiesis or productions of RBC, WBC takes place in the red bone marrow. In early development, it takes place in the yolk sac of embryo, that is in the liver, spleen, thymus, and in later stage, it takes place in the red bone marrow, which is the major site for productions of red blood cells. Connective tissue located, which is located in the spongy bone tissue. So it is the primary site for hemopoiesis. So about 0.05 to 0.1% of the red bone marrow cells are called pluripotent stem cell. So this red bone cell says majority of 0.05 to 0.1% are pluripotent stem cell. So we'll see how the process, by which process they are formed. So now you have a red bone marrow, red bone, we have a red bone marrow. So from that, the pluripotent stem cells which are present in the red bone marrow, they will undergo a subsequent division to form two types of stem cell, that is myeloid stem cells and lymphoid stem cell. Next, this myeloid stem cell, they will further undergo division, that is the colony forming unit of erythrocytes. They will undergo division to form proerythrocyte. Proerythrocyte will undergo further divisions to form reticulocyte, which are called immature erythrocytes or red blood cells, and then they form erythrocytes or RBCs. In the similar way, the colony forming unit of megakaryocytes, they will form megakaryocytes which are immature, and then platelets. Similarly, it is applicable to eosinophils, basophils, neutrophils and monocytes. And the lymphoid stem cells, they are responsible for productions of T lymphocytes, B lymphocytes and natural killer lymphocytes. So these T lymphocytes and B lymphocytes are a class of, they come under the classifications of lymphocytes, that is WBCs. So in that we have sub classification as T lymphocytes and B lymphocytes. So we have already yesterday covered T lymphocytes help in the immune mediated response, and B lymphocyte, they help in the cell mediated antigens response. So next we'll see the process of erythropoiesis, that is erythro means RBCs, poiesis, that is formations of RBCs. So erythropoiesis, that is formations of RBCs, or which are also called as erythrocytes. So erythropoiesis, it is the productions of mature erythrocyte of peripheral blood which take place in the bone marrow with a precursor cell called proerythrocyte. So this is the first, you can see over here, proerythrocyte blast. Proerythroblast, they will divide several times. That is, they will undergo mitotic division several times, producing cells that begin to synthesize hemoglobin. So this is a protein consisting of heme and globin part. We'll see subsequently what is hemoglobin is. So then it will undergo various division. These proerythroblast cells, they will undergo several divisions and then they form mature erythrocyte. So cell near the end of development squeezes or it ejects its nucleus. Remember this, it ejects its nucleus and become reticulocytes, which are immature red blood cell. So red cell production is influenced by one hormone which is called erythropoietin. This is the hormone that regulates the productions of RBCs. Erythropoiesis activity in the body is regulated by erythropoietin, which is produced in response to hypoxia. So now what is hypoxia is? It is a condition where oxygen is deficient in the body. So the principal site of erythropoiesis production is kidney. So from where this hormone is produced? It's a kidney. So we'll now see the various steps in erythropoiesis, or how from red bone marrow the mature erythrocytes are circulated in the blood. The colony forming unit of erythrocytes, that is CFUE, they differentiates into proerythroblast. So we have a red bone marrow, from that pluripotent stem cells form. From that we have two different types of cell, myeloid and lymphoid. So myeloid stem cells, they will differentiate into proerythroblast, which is the earliest phase of erythrocyte series. The nucleus is spherical. It is centrally located. The nucleus is spherical, centrally located, and shows a uniform chromatin pattern. So you can see the chromatin pattern, the fiber-like structure which are arranged. After undergoing a number of mitotic division, this proerythroblast will give rise to basophilic erythroblast. So from here they undergo various types mitotic division to form the basophilic erythroblast. Basophilic erythroblast, these are smaller than proerythroblast. So cytoplasm, it exhibits intense basophilia, which indicates further increase in number of ribosomes. Next phase is polychromatophilic erythroblast. So they will again undergo this basophilic erythroblast. They will undergo mitotic division and produce cells in which hemoglobin is present in sufficient quantity. So when you go from up to above, it undergo mitotic division and the content of hemoglobin just keep increasing. So from here, if you are pro-erythroblasts, they will have somewhat less hemoglobin content because the cells are not fully developed. So when you go from pro-erythroblast phase to erythroblast, the contents of hemoglobin will be at its peak. The nucleus of polychromatophilic erythroblast shows a dense chromatin network with a coarse chromatin masses giving a characteristic checkboard appearance. Next is orthochromatotic erythroblast. So in orthochromatic philic erythroblast, it will undergo number of mitotic division with gradual increase in basophilic philia and increase in amount of hemoglobin. So hemoglobin content keep increasing. Last we have reticulocytes, which are immature erythrocytes. They are not fully mature. The reticulocytes contain the remnant of Golgi apparatus, mitochondria, and few free ribosomes. So once this reticulocyte, they eject nucleus. They eject nucleus. And then once the nucleus is released, the reticulocytes are released from red bone marrow. So once they are released from red bone marrow, they will enter into the circulation. That is, they are ready for their function. They are now mature and circulated into the blood vessels, blood capillaries within 24 to 48 hours. So once they are fully mature, they have ejected the nucleus. Within 24 to 48 hours, they will enter into the blood circulations and carry out their function, that is transport of oxygens and transport of nutrients from GIT, then exchange of gases like carbon dioxide is exhaled from the lungs, then whatever waste is being generated, that is also being excreted. So it will carry out their function once they are fully mature. Next is regulation of red blood cells, that is production of role of erythropoietin. So we'll see now the hormone erythropoietin, which is produced by kidney, what is the role. Now we have seen the function of blood, that is transport of oxygens, nutrients, other substances and exchange of gases. So imagine, or in case if there is any conditions where there is a heavy loss of blood during accident or any injury, there is heavy loss of blood. So the amount of the red blood cells will also be decreasing. So the stimulus that disturb the hemostasis by decreasing this RBCs. So there is a stimulus, there is some disturbance in the homeostasis. By decreasing the control condition, the oxygen delivered to tissue, that is oxygen, amount of oxygen that blood used to carry to specific cells and tissue has decreased. So now receptors, in this case the kidney cells, they will get a signal that there is some disturbance in the body and RBC production has decreased. So also the amount of oxygen delivered to tissue and cells has also decreased. It receives the input. What input is it receiving? It is detecting the low oxygen level. So it is sending a signal to whom? Control center, that oxygen levels in the body are decreased. So you have to send a signal to a effector to produce more erythropoietin, which is responsible for this productions of red blood cells, regulations of red blood cell. So this information is then receptor receives the information that low oxygen levels are there. So control center, which is a brain, will receive this signal. It will tell pluripotent cell, stem cell, or then against the myeloid stem cell, that pro-erythroblast in red bone marrow, which are there uh, to more quickly to transfer the like pro-erythroblast we then convert into basophilic. Then we have a what we have is pro-erythrocytes are immature. So now they have to get converted into basophilic. Then we have a polychromatic erythroblast. Then we have a orthochromatic erythroblast, and then they get immature into reticulocytes, and then mature erythrocytes are formed. So it is telling the control center to make this process faster. That is conversion of pro-erythroblast to reticulocytes or erythrocytes. Once this process is very quick, that is means the process is very fast, more amount of RBCs are available for now. More RBCs are now available for circulations in the blood. So sufficient amount of oxygens that will be delivered into the cells and tissue. So the effector, large number of red blood cells in circulation. So once the sufficient amount of RBCs are available, so there will be increase in oxygen which is delivered to the tissue. So homeostasis is maintained. It comes to normal position. That is from decrease, now it is normal to now we'll see the general characteristics of this RBC. So these RBCs are biconcave disc with a diameter of 7 to 8 micron. So once it ejects the nucleus, the diameter become biconcave disc, as you can see. Then in females we have 4.8 million microliter of RBCs in female and 5.4 million microliters in male. The plasma membrane of RBC is strong and flexible. So because of this characteristics, or because of this feature, whenever they are passing into the blood vessels, capillaries, it allows them to deform their shape without rupturing, as they squeeze to narrow capillary. So when they are circulating in the body from the blood vessels to capillary, they tend to collide to their walls of the blood vessels, or you can say capillary. But still, because of this feature, that is flexibility, they don't get rupture. They remain their shape. So that is they remain their normal shape, that is biconcave. Next, glycolipid in the plasma membranes. It help the glycolipid which is present on the plasma membrane. There are something called as a antigen on the RBC. So now you all will be studying about the antigen when we will be doing the ABO blood grouping and RH factor. So it will be covered like how antigens are bind on their respective surface and how this grouping of the blood is done. So mature RBCs, they lack nucleus because, as you have seen, at the reticulus, they eject the nucleus and they become the reticulocytes. So nucleus and other organelles, they neither reproduce nor carry out any extensive metabolic activity. The cytosol contains the hemoglobin molecule. So each RBC contains 280 millions of this hemoglobin molecule. So now, since there is no nucleus in the RBCs cells, so all the internal space which is there is available for oxygen transport. Even the lack of mitochondria, there are some organelles also, they are not there. So mitochondria is not there, so it will generate their energy, that is ATP, anaerobically, that is without oxygen. So the shape of the RBCs, that is biconcave disc shape, it facilitates its function, that is greater surface area is available for diffusions of molecules, that is diffusion of oxygens and carbon dioxide is available because the surface area is greater. Moving on to a part of hemoglobin. So hemoglobin, it helps in the oxygen transport by binding to iron molecule. So far we have been studying about the functions of blood, that is to transport oxygens and carbon dioxide from lungs to for exhalation. So this function is carried out by hemoglobin. So hemoglobin, it is a protein that transport oxygens in human body from lungs to tissue of the body, and carbon dioxide from tissue of the body to lungs and for exhalation. So this function is carried out by binding to iron molecule. It also transports about 23% of the total carbon dioxide by binding to amino acid and waste product for metabolism. Hemoglobin plays a role in regulations of blood flow and blood pressure. So this hemoglobin, it is divided into two parts, that is heme and globin. So this heme group, it contains an iron atom, as you can see over here, iron atom, so that is able to bind to one oxygen molecule. So these heme portions on which oxygen binds, so on this heme portion the oxygen bind. The next portion of the hemoglobin is globin. It consists of protein called globin. So this globin portion consists of 2 alpha and 2 beta chain. So 2 alpha chains and 2 beta chain. A ring-like non-protein pigment called heme bound to each of these 4 chains. So this is just at the center of each ring, heme ring is the iron. At the center of each of these 2 chains, there is a iron portion. So synthesis of this hemoglobin, it begins in the proerythroblast, as we have seen. From proerythroblast, it starts a synthesis of hemoglobin. So as they are becoming mature cells, the content of hemoglobin keeps increasing and even continue in the reticulocytes of the blood cells. Therefore, when reticulocytes leave the red bone marrow and pass into bloodstream, they continue to form minute quantity of hemoglobin for another day or so until they become fully mature cells, that is erythrocytes. So we'll now see how the synthesis of hemoglobin begins. So as we have seen now, its synthesis begins in the proerythroblast and continues in the reticulocyte stage of red bone cell. So the 2-succinyl choline will bind to 2 molecules of glycerin in the Krebs metabolic cycles to form pyrrole. Now, the 4 pyrrole molecules they combine to form, they will form something called protophorin 9, which in turn combines with this protophorin 9 in turn combine with the iron to form heme. This heme portion, which in turn combine with the long polypeptide chain to form hemoglobin chain, which consists of alpha and beta subunit. So that is 2 alpha and 2 beta chain will form hemoglobin. So depending upon the amino acid compositions of this polypeptide portion, there are various types of hemoglobin chain. So these chains are designed as alpha chains, beta chain, gamma chains and delta chain. So in that, first we have hemoglobin A, which is the most common form of hemoglobin in adult human being. So it is a combination of 2 alpha chains and 2 beta chains, which I have seen in earlier slides. Oxygen carrying capacity of the hemoglobin. So there are 4 hemoglobin chains in each hemoglobin molecules, and each chain has a heme has a prosthetic group which contain an atom of iron. So since there are 4 hemoglobin chains, there are 4 iron atoms in hemoglobin molecule, and this iron atom can bind loosely with one molecule of oxygen, making a total of 4 molecules of oxygen. So one molecule of oxygen will bind to one molecule of iron, which will be transported by each hemoglobin molecule. So this type of hemoglobin change in the hemoglobin molecule, it determines the binding affinity of hemoglobin for oxygen. So if there is any abnormality or any disturbance that will alter the physical characteristics of this hemoglobin molecule, it will lead to various diseases like sickle cell anemia. So we'll be studying about the sickle cell anemia when we are going to do the portion that is anemia. So in what happens sickle cell anemia, the amino acid valine is substituted for glutamic acid at one point in each of these 2 beta chain. So we'll be studying about the sickle cell anemia when we are going to do anemia. So we'll be studying about this anemia in our next lecture. So yeah, thank you.